Here is an overview of clinical laboratory hematology. So by definition, hematology is the study of blood cells. For blood volume, the human body contains approximately 5 liters of blood. Our functions of blood is it transports oxygen from the lungs to the tissues. It removes carbon dioxide from tissues to the lungs as well. The components of the blood is we have a liquid portion of blood, which we call plasma, and then we have, the red blood cells and everything else, which we'll get to later. Erythrocytes. So red blood cells are also called erythrocytes. They are responsible for that oxygen and carbon dioxide transport. Then we have leukocytes, also known as white blood cells, and they are part of the immune system to help try to fight infections in the body. And then we have thrombocytes, which are also known as platelets, and they are involved in our clotting of the blood. So medical lab professionals can stain, count, analyze the appearance, the phenotype, and genotype of the 3 different types of cells that we just talked about, and can help predict and diagnose diseases. So a little bit of background history. We've got back in 1657, Kircher was the first person who described worms in the blood, and those actually ended up being our red blood cells. And then we have in 1674 Van Leeuwenhoek, and he provided an early detailed account of what red blood cells actually look like. Then in the 1800s we have Bezorzero, and he described platelets as petite plaques. In 1902, we have Wright, who actually developed the Wright stain, which you will hear about later on in different lectures, and that actually helped to revolutionize microscopic examination of the blood cells. So some characteristics of red blood cells or erythrocytes: they are non-nucleated, so they do not have a nucleus, and they are biconcave disc, so they kind of look like a donut without the hole, so it just kind of goes down into a little divot. Contents: they are filled with hemoglobin, which is a reddish protein, so that's where our reddish color comes from. In size, they are, diameter ranges from about 6 to 8 microns, and they have a characteristic zone of that central pallor, which is that lighter area in the center, which is kind of like the middle of our, if you want to call it a donut, I guess. It just makes it, no hole, of course, but central pallor is kind of where that is. Functions, again, so it transports oxygen and carbon dioxide, also transports glucose, fats, and proteins, and it can also carry waste products from tissues to the liver and kidneys for elimination out of your body. This is just a diagram of the different types of red blood cells and white blood cells. So we have some terms in hematology. So counts is the measurement of the number of blood cells. So either we're looking at, white blood cells or red blood cells. Anemia, it's a condition that is characterized by a decreased number of red blood cells or low hemoglobin levels, and that can lead to a reduced oxygen carrying capacity. Polycythemia, it's a condition that is marked by an increased number of red blood cells, and that can actually thicken the blood and affect circulation. Hemoglobin assay, it's a test that measures the amount of hemoglobin in the blood to assess oxygen carrying capacity. And with that hemoglobin assay, we usually will use a thing called Drapkin's reagent, and it is a chemical solution used in hemoglobin assays to convert hemoglobin into a measurable compound. Hematocrit assay will measure the percentage of blood volume that's occupied by red blood cells, so sometimes it's called the packed cell volume. Then we have indices, which are calculated values that describe the size and hemoglobin content of the red blood cells, and those indices are MCV, MCH, and MCHC. And don't worry, we'll talk about all these things later, so don't get too worried here. And then we have reticulocytes. They are immature red blood cells. Their count indicates bone marrow activity and red blood cell production rate. So red blood cell counts historically, the method was to do a visual red blood cell count. It was performed using a microscope and a cell counting chamber called a hemocytometer. And then finally came automation. The first automated cell counters were introduced back in 1958. The Coulter principle, this is what many modern hematology instruments use now. It's called the Coulter principle, and this measures cell counts by detecting changes in direct current electrical impedance as the cells pass through the aperture. So clinical uses of the red blood cell count, it's essential in diagnosing anemia, polycythemia as well. If you're in my class, this is a video on how the Coulter principle actually works. For hemoglobin concentration, see principle. So the measurement relies on converting various forms of hemoglobin in the blood into a single stable compound called cyan-methemoglobin. In the procedure, there's an addition of the Drapkins reagent that contains potassium ferricyanide and potassium cyanide, which then react with the hemoglobin in the blood. This reaction actually oxidizes hemoglobin to methemoglobin and then converts it to cyan-methemoglobin, which is a stable and soluble pigment. From there, it has a photometric measurement. So the sample is placed in a photometer and light absorption is measured at 540 nanometers wavelength. The amount of light that is absorbed is directly proportional to the hemoglobin concentration in the sample. So some advantages is the method is highly specific and reproducible. Cyan-methemoglobin is stable, and that allows for accurate and reliable measurements. Automation, so modern hematology analyzers use automated versions of this same technique, and sometimes they will use a modified reagent or detection wavelength to help improve speed and accuracy. For hematocrit, like we mentioned earlier, it is the ratio of the volume of red blood cells to the total volume of whole blood. So the normal values, it's usually about 50%, meaning about half of the blood volume is composed of red blood cells. Alternate name, like we mentioned earlier, it can also be called a packed cell volume. How we measure hematocrit, there is a manual method and an automated method, but the manual method, blood is collected in a capillary tube and then centrifuged. The centrifugation separates the blood into layers, and that allows direct measurement of the red blood cell layers proportion relative to total blood volume. The automated method, so modern hematology analyzers calculate hematocrit by measuring red blood cell count and mean corpuscular volume, or that MCV, and that provides rapid and accurate results. Some clinical importance for hematocrit. So hematocrit levels help diagnose conditions like anemia or polycythemia. Again, if you're in my class, this will be a video on how to do that spun or manual hematocrit test. All right, and then we have the indices. There are 3 of them. The first one is the mean cell volume, or the MCV. This reflects the average size or diameter of the red blood cells that are being tested. It can be visually estimated by examining the red blood cells on a right-stained blood smear. The larger MCV will indicate macrocytic cells, which are larger red blood cells than normal, and a smaller MCV will actually indicate microcytic cells, so actually smaller than normal red blood cells. Then we have the MCH, which is the mean cell hemoglobin. This represents the average amount of hemoglobin or mass per red blood cell. It indicates how much oxygen carrying pigment each red blood cell contains. And then we have the mean cell hemoglobin concentration, or the MCHC. This reflects the concentration of hemoglobin within a red blood cell. It's related to the intensity of staining or the degree of the central pallor in the red blood cells. So a low MCHC can suggest that you have a hypochromic or a pale cell, or normal or high MCHC indicates normochromic or even hyperchromic cells. The indices can reflect the degree of variation in red blood cell volume within a blood sample. So visual identification seen on a right stained blood smear, you can see the differences in the red blood cell diameter. This variation in size is called anisocytosis. The measurement for that is the variation in red blood cell size is regularly reported by automated cell counters, often as a RDW, or red cell distribution width. Again, another video for you. So red blood cell morphology, the method. So we use light microscopy with a right stained blood smear to examine those red blood cells, so microscope. The assessment of it, we are going to visually assess these, and we are going to be looking at the size of the red blood cells, the color of the red blood cells, the shape, are they normal? Do they have some weird shapes going on? Do they look like little balls, like spherocytes? We're going to look at the cytoplasmic inclusions as well, so things like basophilic stippling and Howell-Jolly bodies, which don't worry, we haven't gotten to yet, so we'll get there. But just FYI, that's kind of what we're looking for. Automation, so automated hematology analyzers can evaluate red blood cell morphology using this advanced imaging. However, visual examination under the microscope is actually essential to confirm abnormal findings reported by the instruments, because sometimes instruments can get it wrong. So reticulocytes, these are young red blood cells that contain residual RNA. On a right stained blood smear, reticulocytes comprise about 0.5 to 2.5% of red blood cells in a normal blood collection. These are larger than mature red blood cells, exceeding an average of 7 to 8 microns in diameter. They're also going to stain a little different. They're going to stain slightly blue-gray, and that's due to the RNA content, which is also called polychromatophilic erythrocytes. So they'll kind of stain, instead of that pinky red color like a red blood cell, they're going to kind of stain like a light blue grayish color almost. So clinical significance of reticulocytes. So the presence and number of reticulocytes indicate bone marrow activity and regeneration. Increased reticulocytes can suggest active red blood cell production, such as after someone has lost a lot of blood or in response to anemia. For measuring reticulocytes, you can do a manual method. Reticulocytes can be differentiated and counted by, on the microscope. This will require a methylene blue stained blood smear, which actually will help highlight the RNA in the reticulocytes. As you can see in that picture below, the arrow is pointing to a reticulocyte, so you can kind of see how it's a little bigger than the red blood cells around it, and it's kind of that little blue grayish color more so than the red ones around it. Then there's also an automated method. So we have modern automated, hematology analyzers, and they will actually provide an absolute reticulocyte count, which is the total number of reticulocyte counts. And they will also do an immature reticulocyte count, which gives additional insight into the bone marrow activity and erythropoiesis. White blood cells are leukocytes. So their structure, they do have a nucleus, and it contains the cell's genetic material. Shape and size of white blood cells will vary among the different types. We have neutrophils, lymphocytes, and a whole bunch of different ones. So it's just going to kind of vary on which one we're looking at. As far as cytoplasm is concerned, they usually will contain organelles and granules. The appearance can be granular or agranular, and that helps to classify white blood cells into granulocytes, which would be neutrophils, eosinophils, and basophils, or agranulocytes, which are going to be lymphocytes and monocytes. For white blood cell count and measurement, so for the count, that is the measurement of the number of white blood cells in the blood. Leukopenia, it means it's a decreased white blood cell count, which could indicate infection, bone marrow disorders, or immune deficiencies. Or leukocytosis, which is an increased white blood cell count, and that is often seen in infections, inflammations, or even certain leukemias. To measure white blood cells, we have a manual and automated. So the manual, we can do a cell count performed visually using a microscope and a hemocytometer. For the automated, the analyzers can provide a rapid and accurate total white blood cell count. For the differential count, this is going to be performed visually with a right stained blood smear to identify and count the different types of white blood cells, like neutrophils, lymphs, monos, eosinophils, and basophils. And it provides information on the relative proportion of each white blood cell type. Again, these are the different types of leukocytes, and we will get into each of these later on in the course. These are pictures of each of those. And then we move on to platelets. So platelets, or thrombocytes, they are formed from megakaryocytes, which are located in the bone marrow. Their size, they are small cell fragments that measure about 2 to 4 microns in diameter, so these are smaller than red blood cells. Their function is they play a crucial role in preventing and stopping bleeding by participating in blood clotting or hemostasis. To count or measure platelets, the measurement of the number of platelets in blood, so we have thrombocytosis. This is an increased platelet count, which may increase the risk of clotting disorders, or thrombocytopenia, which is a decreased platelet count, which can lead to bleeding tendencies. Now back to measurement. So we can use light microscopy. So platelets can be counted manually using a hemocytometer, but identifying them can be a challenge because they are so small. And then we have phase microscopy. That's a preferred manual method as it can enhance the visualization of platelets. And then we have automated, of course, the instruments that can perform a rapid and accurate platelet count. It can also provide a mean platelet volume, which is an indicator of platelet size and activity of the platelets. So testing in the hematology lab, we have a test called a CBC, or a complete blood count. The components of the CBC is we look for the total white blood cell and the total red blood cell count. We're also going to be looking for the hemoglobin and hematocrit, and they'll also be looking for the indices, and those are going to be given, from your instrument, so your MCV, MCH, or MCHC, and also the RDW. It can also do an automated differential on the analyzer, so sometimes it will, give that to you, but sometimes we have to actually do a manual one. So when we do that, we will count and identify 100 white blood cells on a right stained blood smear, and we do that to detect and classify white blood cell abnormalities. Same thing with red blood cell morphology. We'll examine the red blood cells for any abnormalities in size, color, and shape, and if there's any cytoplasmic inclusions that we have noticed on the smear. For the blood smear examination, so we have the preparation for it. We're going to prepare a wedge prep blood film on a glass microscope slide. We're going to stain the smear using right stain, and that's going to help highlight cellular components. The estimates, we're going to perform visual estimates of platelet and white blood cell counts using a microscope, and that's just kind of to double check that the instrument's correct. We're just kind of doing a double check there. For cell morphology, we're going to examine white blood cells, red blood cells, and platelets, and we're going to be looking at their size, shape, color, and if there's any inclusion bodies or any abnormalities. Again, another video on how to do the wedge prep slide. All right, so then we have the endothelium of blood vessels. This is the location. It lines the inner surface of the blood vessels. The function is to maintain normal blood flow by providing a smooth, non-thrombogenic surface. It provides attachment sites for those platelets during blood clotting, and it also enables white blood cells to move from the bloodstream into surrounding tissues, which is a process called diapetesis or extravasation. Then coagulation or blood clotting. The definition of this is the process of changing blood from a liquid to a solid or semi-solid state to stop bleeding. The system involved with this is the coagulation system and platelets. The coagulation system is a series of enzymatic reactions that lead to a fibrin clot formation. Platelets have cell fragments that initiate clot formation by aggregating at those injury sites. We have the vascular system as well. That is the blood vessel walls contribute by constricting and providing a surface for clotting. And then the fibrinolytic system, this is actually responsible for the breaking down of the clots once healing is complete. So a test we use to monitor coagulation is a PT, or a prothrombin time. This measures the extrinsic and common pathways of coagulation. Then we have the PTT, our partial thromboplastin time, and this measures the intrinsic and common coagulation pathways. And we also can do a fibrinogen assay, and this measures the fibrinogen levels, which is a protein that's involved in clot formation. Once again, we're going to talk about all these in detail later on in the course. This is just kind of a high overview at the moment. All right, so additional hematology procedures, they can do bone marrow assays. The purpose of this is to analyze immature blood cell precursors to help diagnose and monitor certain blood disorders. So we have different types of bone marrow samples. We have aspirates, and these are liquid samples that are drawn from the bone marrow, and then biopsies, which are actually a solid core sample of bone marrow tissue. Then we have cytochemical stains. These are used to differentiate and identify abnormal myeloid, erythroid, and lymphoid cells by highlighting specific enzymes or cellular components. Some of these common stains we will use are myeloperoxidase stain. This helps to identify myeloid lineage cells. Sudan black B stain, that helps to stain the lipid-rich granules in the myeloid cells. Esterase test, this can differentiate types of leukocytes based on esterase enzyme activity. Periodic acid Schiff stain, that highlights the glycogen and other carbohydrates in cells. Then we have acid and alkaline phosphatase stains, and this can detect specific enzyme activities, which are useful in classifying leukemias. Some other additional testing we can do is flow cytometry. This is quantitative, and it measures the number of specific cell types. And it can also be qualitative, and this uses laser-based technology to detect the presence or absence of specific cell markers or proteins. You can also do molecular testing, which can detect abnormalities associated with hematologic diseases. And then there's the Philadelphia chromosome, which is detected by molecular testing. This is a chromosome that is on chromosome 9 and 22. This chromosomal abnormality is diagnostic for chronic myeloid leukemia. The detection is important for diagnosis, prognosis, and treatment planning. And once again, we'll talk about all this in more detail later. Some additional testing, once again, we have osmotic fragility test. This is used to diagnose spherocytosis, and it does so by measuring red blood cell susceptibility to hemolysis in hypotonic solutions. The glucose 6-phosphate dehydrogenase, or the GCPD assay, this detects red blood cell enzyme deficiencies. It's important in use for diagnosing the GC or G6PD deficiency, which is related to hemolytic anemia. We have the sickle cell solubility assay. This screens for, you got it, sickle cell anemia, and it does so by detecting hemoglobin S. Hemoglobin electrophoresis can identify inherited hemoglobin abnormalities like sickle cell or even certain thalassemias. Then we have the ESR, or the erythrocyte sedimentation rate, and this measures the rate at which the red blood cells settle in a tube over time. This can indicate inflammation or other disease processes. Body fluid assays, so the analysis of these fluids such as synovial, peritoneal, pleural, and cerebrospinal fluids are used to help detect infections and other abnormalities. All right, so quality assurance and quality control. So quality assurance is a systematic process that is used to determine whether a product or service meets specified requirements consistently, whereas quality control is the process of measuring and testing products to ensure they are uniform and meet established standards. So the function of this, both a QA and QC are here to ensure laboratory results are accurate, reliable, and correct by maintaining the integrity of the diagnostic testing. So very, very important in any department of the lab, not just hematology.